So, good evening to one and all. It gives me pleasure in welcoming you all for the endowment lecture on Are We Ready to Feed 10 Billion People? Challenges and Opportunities by Dr. Sambat Perumal, Technical Lead Plant Improvement Program, Global Institute for Food Security, University of Saskatchewan, Canada. Now, I request Dr. John Joel, Professor Biotech, to welcome the gathering.
Good evening, one and all. So it's a great pleasure for me to welcome Dr. Sambat Permal for this endowment lecture. Regarding Sambat Permal, he is our alumni. He did his master program in biotechnology during 2006-7, and he did his PhD program at the Seoul National University, South Korea, and during 2009 and 2009. And later he did his again a postdoctoral program there at Seoul National University during 14 and 18. And then he joined as a postdoctoral research again as Agricultural and Agri-Food Canada. He got the MITCAS fellowship. That was a prestigious fellowship in Canada at the University of Saskatchewan. And he became the research officer in during 2021 at the University of Saskatchewan. Then later, during current program, he became the research associate and became elevated as a team lead for the plant improvement program. So he was all along working in different crops like canola and this alfalfa. Currently, he is working in prairie crops like wheat grass, then alfalfa, then brassicas, so brassica genome, and he has got wide knowledge in the field of molecular biology and bioinformatics like GWAS, QTL analysis, transcriptome analysis, and this NGS programs and SNP analysis and phylogenetic studies. And with this wide knowledge, he was able to publish papers in peer-reviewed journals, more than 26 research articles he has published in this young age. And he has also written chapters in different books. Now, while I was interacting with him, I was able to find out his the knowledge level in bioinformatics and is really great. And it is the time for him to share his knowledge that he has gained over a period of this time, 10 years of experience that he has gained from Seoul, that is South Korean universities, as well as Saskatchewan University. And he has got funding from lot of funding from different sources. Presently, he is working in the project identification and exploration of genome structural variations for trait improvement in canola and wheat crops. And the present lecture he is going to present is on it's a really a great challenge, and he is going to give you a lecture on how to feed the 10 billion people. That is really a great challenge that we are going to face, and he will open up to the younger minds how to move forward. So now I request Sambat Perumal. Before that, I request Madam Head of the Department, Dr. Uma, Head of the Department of Biochemistry, to welcome him with a bouquet. Okay. So thank you for the opportunity. Now I request Sambat Perumal to present.
Hello, everyone. Hope you can hear me in the back, right? Thank you very much, sir, for your kind introduction. It's really a pleasure to be back here, where I did my master's and see a lot of my new or old friends as well, and seeing my teachers. It's really a privilege, and I'm really happy to be here. So in this lecture, as Dr. John Sir said, I'm going to talk about feeding 10 billion people. What are the challenges we have, and what are the opportunities that how we are going to mitigate or for that to produce crops that feed 100 billion people. Before that, I'd like to give a brief overview, though John Sir already said what was my journey previously. I did my bachelor's from Annamala University, and I did my master's in plant biotechnology from 2006 to 8 in plant CPMB, plant biotechnology. Uh, so during that time, so Cynthia he was my supervisor, and he keep encouraging everyone, including me, to study abroad. So that was the point that so need to do some PhD program abroad. So I ended up searching for opportunities and found that Korean government scholarship program in 2008. At the time, I haven't finished my thesis at all, so it was very hectic time. And Cindy Saris was kind enough to, help out and finish my thesis. I was defending my thesis the day before flying to South Korea because it was mandatory. I had to take that flight. If not, like I had to cancel the fellowship. So I had to I had to thank this time. Remembering this moment, I'm thanking Cindy sir for that. So once I finished my PhD in South Korea with the fellowship program, it was really a good opportunity to go, explore the other space. I worked on plant bioinformatics, plant genomics and bioinformatics mostly. The brassica is the one of the important crop in Korea as well, the cabbages. So that was I mostly worked on. And in Canada, canola is one of the crop which is very economically important crop. It's kind of related to my PhD research program as well. So it was a good opportunity to go to Canada, explore and expand my knowledge as well. So during 2016 to 19 as a postdoc, my second postdoc, I got a fellowship through MyTax, which is an industrial fellowship. It was a really nice program. You can have so industries funding for your study, like 50% of them to do like things that's really needed, right? So that's one of the thing even here the industries can consider doing that. So then it's things progress. I'm still working on learning more and more and keep progressing. Currently, I'm a technical lead for the plant improvement program, where we have like multiple projects for improving crops, including canola, wheat, and forage crops like alfalfa, wheat grasses. So our institute is a Global Institute for Food Security, which is which is present in the University of Saskatchewan, jointly funded by Nutrien, which is a company, and then government of Saskatchewan. It's a provincial, like a state government, and then University of Saskatchewan. This is all three agencies come together to provide innovative solutions. So you discover the problem and develop solutions and deliver to people or it's needed. So that's what it's just kind of thing organized. They kind of donate like 10 million each. So to have this kind of set up an institute. So this our institute where we work towards providing solutions to the problem that's global wanted, as at the aim of providing food security to everyone in the world. So I just try to highlight some important things, like we have this platforms, the multiple platforms to handle different areas. For here, I show the sequencing platforms, the current sequence packed by on the nanocore, and then high throughput phenotyping. I've just shown the drone ones, but they have like big machines to phenotype in a really high throughput manner. So that's what is they are like kind of adopting the new technologies, try to explore and do it more efficiently as possible. We have this multiple platform in our institute. So Opal is a sequencing platform and cell biology, where you do this plant tissue culture, double haploid population production, plant transformations, and the bioinformatics team and the greenhouse team. So they come together, work together, how we can collaborate each other. So they have built such programs and for providing solutions. For Gibbs, so we have multiple, other than platforms, we have multiple programs like accelerated breeding, biomanufacturing, policy and regulatory. So I belong to the green one, that accelerated breeding, where you work towards to identify, so like gene discovery or develop new genomic resources, or and have those knowledge, like for example, doing the GWAS to identify the target genes, provide to the other team, they do that gene functional characterization. As I said, our team works on genomics and mostly phenomics for crops like wheat, canola, and forages to develop climate smart crop. So we have like 8 different programs running. We acquired like around 2.5 million fundings for the next 4 years from provincial, which is state, and federal and industrial programs. Industry also provides funding for research programs. So my job is I worked to co-lead a couple of projects, and I also oversee technicians and postdocs and the PhD students that's in our program to run the projects. Okay, coming to today's matter, I'm going to talk about this feeding 100 billion people, the challenges and opportunities. These are the outlines for today's presentation. I like to give a brief introduction about so what's the why or what's the need for food, and what the major challenges in producing those food, and what kind of opportunities we have, especially in terms of genomics. Okay, as everyone aware that we are growing, human populations are growing and more, and it's expected to reach 10 billion, almost 10 billion by 2050, which is which means that there is a demand for food. With the current pace of food production is not adequate. So we need to increase the food production without expanding any arable land. Even the fact that we are losing our arable land due to various factors that's I'm going to cover later. This through conventional breeding approaches, the rate of genetic gain is quite the standard, but it's compared to the need, we say it's quite low. So with the 1% increase, it's not going to be enough. So there is definitely a need, the urgent need for improving existing crop, higher yields with better nutrition, and considering that climatic change, we have to provide that climate smart crops. So I see in the graph that the total population of India is reaching almost 1.6 billion, and it's about to reach about the peak in a couple of decades. The United Nations has developed 17 Sustainable Development Goals aimed to achieve by 2030. One of them is zero hunger. Set the top goal. The second goal is mentioned here. All the United Nations all the 193 members agreed to end poverty, ensure prosperity, and protect the planet as well. However, still 1 in 10 people are still suffering from hunger. So we are still a long way to go to feed those all the people, even the current population. So challenges. What are the challenges we have? So I just kind of listed 4 challenges here, but there are still many more challenges as well. But considering this presentation, I'm going to talk only about these 4 challenges. First one is the global warming and climatic changes. As everyone aware that we are global is that the atmospheric temperature is getting hotter and hotter is due to the heat trapping pollutants that's present in the atmosphere. It's it's the solar radiation get reflected to the space, but due to that greenhouse gases, especially carbon dioxide, methane, nitrous oxide, which have the tendency to hold the heat, which makes that atmosphere is quite hotter. So this effect is called a greenhouse effect. So this has a significant impact on the agriculture as well. It causes increased frequency of weather extremes, drought, storm, flood. That's we are experiencing. Global warming causing long-term impact on agriculture. So we need to act on that to avoid any more dangerous. Even recently, United Nations has announced code red for humanity. If you're not going to take care of this global warming issue, By the end of this century, we are expecting to increase the global average temperature by 4 degree, which means that it can submerge that significant part that present in an island. So one of the important cause for global warming is CO2 emissions. CO2 emissions in India is about like 1.8 metric tons since 2019. It's primarily due to this electricity and the heat generations. However, farm sector also cause like 19 to 25% of CO2 or greenhouse gas emission. For example, rice is uses or emits like 20 to 40 kg of CO2 to produce per kg of rice. Um, so here is a literature showing that different food groups in India, how much CO2 is emits per kg or per like 1,000 calories. If you see in the bottom um, of the figure. Uh, yes, of course, rice are frequently flooded during growth, which generates methane emitting bacteria. That's one of the reason. However, like now perennial rice is been getting more popular, and which can reduce that even input cost and also even increase the carbon sink as well. So that's also we can consider. So carbon footprint from agriculture, yes, is mostly from soil management, as I said. It's about 292 kilodalton, uh, though there are other causes like livestock or manure management, energy, and even burning of crop residues. The soil management, especially like primarily through fertilizers, and that causing major and carbon dioxide. There are though there are some positives like a carbon sink, which is stored in the soil, but it's very limited. Due to global warming, drought is one of the issue that's in globally as well as in India too. As you can see in this picture, you see uh, India is in a red, which is like quite high, 40 to 80% will be water stress by 2040. So there is definitely need for like young scientists like you to work towards that goal to produce crops uses less water or like resistant to drought. Yes, even though it says Canada is in still in blue, but still like it experienced a heavy drought. So yield loss can be significant. It can cause so anywhere between like 50 to even like 92% rise is got affected due to this drought. So it's definitely a serious issue that we need to tackle or consider. As I said, like even it's Canada was in blue. Last year we experienced quite a significant drought where usually Canada is groundwater is rich, but they used to be like more rain-fed condition because they just grow just only one season from May or June to September. Um, During that time, almost 30 days without any rain or like more than 30 degrees centigrade, which is a significant period for the crop growth, which causes real damage to the plant. For example, the canola and the spring wheat, that's the major economic crop in Canada. Those grown in 3 different geographies, Manitoba, Saskatchewan, and Alberta. So those are prairie regions. So which got affected like 50%. For example, as you see that canola in Saskatchewan, and we experienced really severe drought that causing 50% yield loss. So it's a big issue. Definitely, we need to consider work more progressively. Uh, so this here is information says especially small scale farmers that gonna be affected really much by the drought. So especially by very soon by 2030, they're like East Northeast or Central America, East Africa, New Zealand, especially in India. So they need to adapt with the better strategies to accommodate this extreme weather conditions. The second important challenge that I like to focus on focus is the latest crop diversity and long-term generation times, which also is affecting to produce crops in a sooner time. So here I show that maize with different colors of maize, which showing the diversity. However, we just kind of selected for domestication and selection. We lost almost a lot of major leagues because we just focused on yield or like some very few traits. That's how we lost almost a major genetic diversity. That's need to be brought back into breeding program. That's I was talking to Dr. John. He was telling that so they're bringing back from gem blossom uh, to decimate those no, important alleles to be like more climate smart crops. That was the interesting thing, sir. Yes, as I said, like we lost like much of diversity due to selection and domestications. Those those circles, like other than that green, yellow and blue, so we have those alleles that may be resistant to biotic and abiotic stresses that may have like more that need to be exploited. So the next one is the long generation times. Conventional breeding approaches, yeah, definitely produced high yielding crops. However, the generation take the line development is taking really a lot of time. For example, when you cross and develop a line, which takes almost 5 to 10 years, depends on the cycle of the crop. However, when you use the recent technology like a speed breeding, you can reduce this line development. This is quite work well for the research purpose, where you can develop finish those line development in 1 to 2 years compared to 5 to 10 years. The next important challenge I like to talk about is loss of agricultural land, especially due to that salinity or human activity or sea level rise. You can see in this picture that the graph shows, especially that blue line, we are reducing that arable land when compared to 1998 to 2016. We lost almost 1.5 to 2% of the arable land, which is quite significant. And on the other hand, you see that populations is been quite it's been increasing. Definitely, there is a lot of land that has been modified for the or there are a lot of human activity that's using the arable lands. Salinity has been one of the major issue in India for a long time. Currently, almost 6.74 million hectare arable land is affected by salinity. This is about like about 0.5% of the total land, which is mainly due to accumulation of chloride and sulfate salts. The salts from groundwater, that can be a cause, and maybe that over application of fertilizers. That's one of the other reasons too. Um, you know that the salinity can cause yield loss up to 100%, as you see in the top right figure. There are a lot of salts, and the bottom table showing that most of the saline affected soils in India. There is a need for better variety that's withstand in heavier salt conditions. Rise of sea level that's due to global warming is one of the big issue too, which can cause that especially the lands and the seashore lands is got affected. Um, so as I said, like if this happens like end of this century, 4-degree increase can cause 2.2 meter uh, um, by end of this 2.2 meter sea level rise, which means that it can cause severe impact. Here on the top, you see that with a 2-degree increase, there are countries that got affected by that sea level rise. Even India on the bottom figure, you see that the blue marks, those are highly going to be affected immediately by the sea level rise. I want to talk about some of the government policies that hinders that producing food crops to feed the 100 billion people. India has planned for the hot carbon footprint to reduce that emission by 45% by end of 2030. which uses the route of generating 50% of electricity from non-fossil fuel sources. And that's a good thing. So we you don't get like a lot of carbon, right? The creation of carbon sink. That's you store carbon dioxide in the land through additional forest and tree crops. So this is where perennial crops gonna be really helpful. Like if we are converting our crops into perennial, that can be very important. Like rice is been there has been successfully developed perennial rice where they attain the yield same as that what you grow, where there are a lot of cost got reduced, like input cost, labor cost, and even it stores a lot of carbon in the soil. If you wanted to achieve carbon neutral, carbon net zero by 2070, there's significant plan need to be done, and we also need to work on as agriculture scientists. We need to work on how we can help with that. So there is a need for 10 trillion investment now to work on that to grow more trees and to achieve this net zero. And GMO policies still it's been controversial for many years. Uh, just recently, India approved just first GM food crops, which is a mustard crop, which is expected to produce 28% of yield. However, there are many crops, many food crops that's still in pipeline that need to be approved, and one among them is golden rice. As uh, so these are things that hinders that develop more better crops. However, I think it's in the right route. I mean, India is in the right route to work on this GMO policy. Okay, I'd like to switch a gear about talking about opportunities now. So what are the opportunities, the challenges I mentioned earlier, especially using the genomics tool? How to use the genomics tool to breed a better crops in a faster way. So that's the kind of an agenda for this opportunity. So for the breeding better crops, there are different tools, that recent tools that help to elevate the better understanding, deeper understanding, and develop high quality markers that help to tackle or help to identify markers with much better precision or accuracy. So that's what I like to give and highlight it. There has been a lot of innovation in sequencing technologies. Sequencing, as you may aware, that's the sequencing of any crop that's using different kind of sequence technology. Illumina was the one predominantly used for to sequence, to generate assemblies, or even do that marker discovery, even characterization like a gene association analysis with the phenotypes information. However, when the long read sequencing, especially the nanopore, which is the middle, and then packed by a sequencer, which are producing longer reads compared to the Illumina. Illumina is producing just like a less than 500 base pair, while comparing to the 10,000 base pair average by packed by a nanopore. Though there are some error rates there, but still can able to achieve really good longer rates. On my right, you see that there's a circular plot which showing comparison between two different sequencing platforms. The one we sequence, this is the Brassica nigra genome, chromosome 6 compared using it was developed using short read on the pink one, that's a short read one, and the blue one, which is developed using long reads. You see the gap in the middle, that those ribbons, like the pink color one in the middle, that connects each other chromosomes, which means those regions are overlapped, so those regions are present in the other chromosome. But in the center, you see that there's a big gap, so that's not able to assemble using Illumina sequence technology. So we have the technology to export these hidden regions where you can get like more gene information. Though this is more of repeat rich, that green color bar, which showing that how much repeat present in this. The lower the ball or length, which means the lower the repeat, higher the bar means the higher the repeats. Using long read sequence technology, there has been lots of work going on. As you see from this blue dots, those are those are really high contiguous, which means like the less gaps, like our gapless assemblies compared to the short read versions. Those are like much fragmented assemblies where you lose a lot of information. Like when you try to compare the genomes, you're still not comparing 100%. You're still losing a lot of information when you use that. So this technology definitely helps to elevate the genome assemblies in a much better way. Say like nowadays we have like telomere to telomere assembly of human genome developed after 20 years. We have the first reference genome. We just developed a whole genome assembly of human. I mean that's a good thing. So we have the technology now. We can develop more good assemblies in the future soon. Um, the second one is the technology about sequencing, or sorry, phenotyping multiple traits in a high throughput manner. So it's called high throughput phenotyping. It's this area has been rapidly improving now, and it produces really good phenotype information. We have tons of genotype information, like a lot of sequencing available. However, we don't have enough phenotype to compare what the genotype information is for. So that's where this gonna be helpful. Even the traits which is really hard to know map or hard to do the phenotyping. For example, root traits that can be done high throughput manner nowadays. So that's one of the important thing that's been really promising. So we can map the traits. For example, the more longer the roots, it can go deeper and get the water. So which means that it can be more drought tolerance, or drought avoidance. So sorry, drought resistance. So you can go, or you can even identify genes that's responsible for that to do the functional characterization. These are the different platforms, imaging platforms to do that imaging of different scale or different types of cells or different types of organs, like a plant cell, seed, leaf, or even the field scale. I use the trait seeker. That's also available in our institute to do that high throughput manner. I mentioned earlier, like we lost a lot of genetic diversity due to selection and domestication. For example, you see that land race questions so far. Am I too fast, or is it okay for you? So in order to capture those missing alleles, that beneficial alleles, there are approaches, obviously like pan genome approaches where you get to sequence or collect the diversity of entire species. For example, here we have the domesticated lines, wild lines, and other closely related lines you can sequence them, catalog those variations to develop a diversity panel, which then be used to understand that the correlation with the phenotypes through genome-wide association studies. So that can be done. So that's where you can able to bring back those important lines into your breeding program, because we have lots of germplasms that you want to bring into breeding program, but it's quite impossible to bring everything into that. So you want to identify the lines which is beneficial. So in this case, the pan genome approaches will be very useful to identify lines with potential interest. One of the recent study on tomato, where they developed a pan-genome level variations using long-read sequencing, because it has been done on short-read as well, but it couldn't able to find much better markers that correlate with the traits. So here they used a long-read sequencing to sequence, especially to identify the structure variant markers. This is the similar kind of work we also working on and in canola, to sequence 50 different lines to develop 50 different reference assemblies. This we generally used to do single nucleotide variant SNPs. However, the structure variants, something like deletion, insertion, tandem duplications, and those insertions, copy number variations, those are hard to predict using short read sequence technology. That's why like we had the technology not quite adequate previously, but now we have this long read sequencing that can go deeper, identify those structural variants. Here, this under different diverse tomato germplasm that used to sequence with the long read sequencing to identify more than 200,000 structural variants, which are like more than 200,000 new variants, new markers. Right. I try to yell as possible. So the structural variants is definitely causing gene expression impact. Most of the genes with the structural variants is reducing the gene expressions, as you see from the gene expression profile on my left. So here, so they found that there are significant traits like flavor trait or fruit size that has been impacted by structural variant markers. So that kind of thing can be done using long read sequencing. Apart from that, there are lots of approaches can be done using multi-omics approaches, transcriptomics. What I haven't mentioned is all genomics, right? So you can do the transcriptomic approaches to understand the differentially expressed genes, or with and the proteomics or metabolomics, and then phenomics I mentioned, like how we are doing like high throughput phenotyping. Yeah, so you can combine as many omics as possible to understand or better target the regions or trait with much clarity. So that's what I like to say. That's we have to include like multiple omics tools. So currently CRISPR, for gene editing, CRISPR is one of the important tool to edit genes in a much faster way. Here, one of the examples showing that wild tomato by multiplex editing multiple genes at the same time, such as like plant architecture, SP, the flowering time, fruit size, they can domesticate those lines. Those lines are more they tend to be resistant to biotic and abiotic stresses, so they wanted to convert them into domesticated lines. So this technology can be helpful in speed up or develop a crops with the way you wanted. So I want to talk about how you produce faster crops, better crops in a faster way. There are two different approaches that can be used using genomics to develop that crops faster. One is the genomic selection, and another one is speed breeding techniques. Genomic selection, where you select best candidate lines. Basically, you eliminate the lines which is not useful. How you do that is you predict that these um, you have the phenotype information and genotype information of some set of lines. You use so that's called a training lines, and the testing populations where you have the genotype information alone, but that's enough to compare them. So when you have this kind of marker set or the haplotypes, then it can perform in this manner, something like that. So you're just predicting them and identifying. This way you can shorten the life cycle. You don't have to wait until the crop to grow and see how it performs. Instead, you just use the small leaf to extract DNA, and then you can see that. So that can be done as well. So this can help rapid selection of superior genotypes. It's definitely it improves the genetic gain, as you see that compared to the conventional breeding approach. This genomic selection approach for oscillator breeding, you see it includes all the environmental variables as well. At the end, it increases the genetic gain. You see the blue lines. It's just 28% compared to this phenotype alone, which is 19%. How you can avoid long-term generation times. As you see that conventional breeding approach, you wanted to pyramid few of the traits. It takes up almost 8 years for this fit. However, when you use the zero greenhouse or even speed breeding, that reduces time to 1 to 2 years. So that's the kind of thing that you can do. So how you develop a what's speed breeding, how you develop that. So using glass houses, so you develop like 3 seasons mm, 3 crops per year. Use a speed breeding where you produce more light. So you see that 12 hours in the normal greenhouse, in the speed breeding you produce 22 hours of light it enhances to mature faster and works faster. It's like you're just turning on the machine for a long time by providing like more lights. So it's such a way that you can produce the rapid cycle plants. For example, wheat you can produce like one crop in 66 days. Such as you can see on the table that there are important crops that how much days to take to develop these rapid cycle crops. Okay, I'd like to finish up with just a few more slides by providing what are the promising outcomes by genomic approaches. One such approach is here, where they alter that photosynthesis time, where plants when there is too much sun, the plant try to protect themselves, so photo protection. However, when the plants is protected by light, you see, so that brings back it's like kind of wake up and then try to do the work again. So that cause be delayed. So when they altered that time, that's the delay in recovery of photo protection. You see the red line. So as soon as that light is done, it wanted to recover the relaxing phase compared to the blue one, which is about like 30% increase in the photosynthesis. So which is increased 33% of soybean yield, just altering few of the xanthophyll cycle genes that speed up the light absorption, and it produces more photosynthesis. More photosynthesis, it produces more yield. So that's the kind of thing you can do using genome editing. And the other thing is in the rice, the one gene overexpressing of DREB gene OS increases yield up to 41 to 68%, which is quite significant. So that's not only increases yield, it also shortened the growth time and improved nutrient use efficiency, especially nitrogen use efficiency, and promoted efficient resource allocation. This also tested on wheat, and I tend to be worked on that as well, like by editing this particular gene, like 22.6% yield increase. One other thing is recently I see our Indian Institute of Rice is Hyderabad. They edited a genome. They developed a yield increase up to 40% in the Samba Masuri, where the grains, the panically from 230 to 400, which is also quite promising. That's everything done through the genomic approaches. First, you have to identify the genes of interest, then edit them. So that's where you can bring them with that the way you wanted. So summarizing my talk, just so are we really ready to feed the 100 billion people? Yes, I think we are getting there, but there are still challenges, like especially the global warming is the important challenge that's need to be addressed. That can agriculture can help to avoid that too. Also, we have to produce a plant that can withstand various biotic and abiotic stresses. Genomics can be a key or important tool that can be adopted with multidisciplinary platform to identify to develop a crops uh, with a much better nutrient use efficiency and biotic abiotic stress tolerance, or higher yield on nutrition. So this is a path to produce improved crops. That what are the tools that I talked on, then to produce the better and faster crops. It's a kind of overlay here where you have a germ blossom in the green, and then you have the different kind of germ blossom. You want to mine the alleles, important alleles, like using pan-genome approaches. You can identify the whole gene species-wide gene information. Then so doing that multiple genotyping or phenotyping or endotyping using currently available tools that also can help we identify genes of interest through QTL or GWAS mapping approaches. Genomic selection and speed breeding can help fast enough, as I mentioned, and CRISPR can be important in terms of editing the genes that you discovered through mapping approaches. So then you can create a improved germ blossom. With that, I like to thank everyone for your kind attention. Sorry for the little breaks. With that, I like to thank organizing committee as well as everyone here for your kind time.
Session is open for discussion.
So what are the challenges of pan-genome data analysis?
Okay, for the pan-genome technology, that's a good question. Thank you. So pan-genome, you want to capture the entire genetic diversity of a species, right? So you want to know what are the species you wanted to include. That's the important challenge. There are close related species can be included in your pan-genome analysis. It's been growing more and more. When you sequence, for example, 10 lines, and the next time you find like you are still going to be missing some traits or some alleles, right? So that's you wanted to add in then more. So that's where you can mitigate that problem, but still a selection of lines, that's how many lines and how rich the diversity is. So those are the things is challenging. And when you wanted to include global diversity, it's going to be really diverse as well. So that I think that's what I feel like it's identification of lines for parent genome. And then technologies wise, like how you want, which technology you want to use, how precisely, how you want to do that parent genome characterization. For now, like long read sequencing, it's not quite cost effective as of now, but I think it still is progressing. Expected to produce more than 5 times that what is producing now. Like for example, you are spending $10,000 now to generate some 500 GB data. Then in less than 3 years, it's gonna be like 5 times, which is like 2.5 terabyte data you develop. And the affordability, so cost wise, like developing nations, is that cost is one of the important factor. We have a lot of resources here, but thing is that like the money or funding for that to do sequencing and thing. For developed nations, yeah, they still have money, but they have priorities, right? So they wanted to work on crops that is really giving you money, or like so that's what I think it's challenging.
Thank you.
Hope that answers your question.
Yeah, thank you.
We are working on it. We have 50 lines being sequenced. I'm doing that. We have developed a genome reference genome for those 50 lines. Those 50 lines are spring types. So spring types is the major type in Canada has been grown, like just less than 4 months old crop. There are different types, spring type and winter type. Also, there are winter types. You grow them, you need vernalization to flower, and then after the winter it grows, right? For my current project, I'm working on with the 50 spring canola line. Canola is a kind of mustard. So we develop reference genome using long read sequencing. We generated almost 50 to 100x coverage data for each line using nanopore sequence technology, and we cataloged SNPs and structural variants on those lines. Yeah, that's the project I like to now wrap up in this year to get it done. We're still working on it. This we wanted to include some of the other omics aspect, like we have so one of the important thing in the long read is you don't have to do methylome sequencing alone. You can use that same data to the methylome analysis, because you can see the methylated C as a different peak. So using that same information, you can do the methylome as well. We are doing that one, and as well as transcriptome as well. We try to do maximum omics as possible, as I mentioned, to get the traits that's not mapped previously. Yeah.
Thanks, Dr. Sampath, for a nice overview of modern breeding approaches for development of improved crop varieties. I have a question. In one of the slides, you mentioned transient expression of xanthophyll genes. Do you have any information on how they made transient expression?
I I don't know, sir. I just yeah, it's about how I read the paper. Yeah, I haven't gone through in detail. Maybe this paper.
It's interesting, transient expression. Genate to express genes transiently. I just wanted to, out of curiosity.
No, sir. Yeah, I haven't. Yeah. Yeah, this De Souza et al. 2020 was a recent one. This work has been amazing actually. They have been doing this. Stephen Long is the one. He has been doing this for almost 30 years. This work here, there are like 3 groups that are working on manipulating this photosynthesis recovery rate. Yeah, he has been like there has been progress being made. However, at the end, like you wanted to bring into commercial like a wide scale, right? It's all done in greenhouse wise, but yeah, this has been amazing. It was recently published though, yeah. This was 2022. Yeah, that will definitely give us some information. Basically, we do genomics and bioinformatics, not that gene functional characterization in our our team. Yeah. We have a project. We have a postdocs working on that, improving canola protein quality. We are characterizing that the lines that I have that's pan genome has been developed, been used for that to test that. Yeah, there is a project now, but still in a preliminary stage. Yeah, definitely. They wanted like industries wanted to improve that quality of oil and like protein too. Protein meal, that's also important for the cattles. Yeah. Yeah, which one? Canola oil is for like a soybean oil, like it's a edible purpose. Yeah. There is a biofuel purpose. There has been research going on, but this is the low glucosinate and low erucic acid. Like what we have the mustard here, it's quite strong. You can't use it much, but there it's like I would say like sunflower or a ground. How you use a groundnut oil here, a similar way they're using. It's quite low fat too, so that's also quite important. Canola is a major edible oil, like what we have here, groundnut or sunflower oil. How far? Sorry. Uh, double Z. Yeah, we have already 00 lines. It's been developed already. Yeah. So those. So 00 is a low glucosinate or like zero glucosinate, low erucic acid. So those are the two acid. That's what called the 00 lines. That's been developed in 1971 itself, but there has been yeah. We have double haploid lines has been produced. We have new programs that's work on developing accelerator breeding, like the traits. We are working on different traits, not that 00, because it's already fixed. That 00 is fixed, and then we are working on different traits. For example, improving more yield or like more resistance and pest and disease ones. So those are the things we are working on. No, it's I I would say it's mostly around that range. You see that even this one, you see on this here, you see the 22 hours light, 22 hours light. Basically, you are trying to give more light to the crop so it can just keep working, only giving like less. There has been modification in the protocol. Lee Hickey is from University of Queensland. He's the one who's been vigorously developing these protocols or even setups that you can, but mostly it's 22 hours. You can. Here I see 22 hours of light. I don't know, it can able to see it clearly. Yeah, it says 22 here. I can't work with mouse. It's not working, but scientist has been developed many protocols for like there's optimization also been done. But yeah, I would say it's close to that. I'm not sure, like 16 hours. I haven't. Yeah. Yeah. You can see differences in the phenotype. Like for example, seed, you can see like it's not a healthy, good seed, but still viable. You can use that seed for next generation. It's not like when you measure the yield, it's not like as usual like what you develop, excuse me, with a normal cultivation method. But still you can see that genetic intactness. So you can that's been working. That's what is proved. Yeah, definitely it's a good. Yeah. Single cell protein to tackle what? Sorry. Um, Okay, I try to answer effective as possible. The first one you asked about single crop. It depends on the need, right? So the wheat and rice are the major, or your corn is the majorly grown because this was needed. Like we don't grow millets for that much big scale. It depends on the need. We will use the diversity. Obviously, like we lost like many crops. We are not They're kind of orphan crops. That's called. We already lost many crops that's not been grown. We are growing only less than 1% of the crops. I would say, even that we have a rich diversity, but we not growing that. So the crops we already growing. Like for example, rice, we still have a lot of diversity around the world. It's not been used properly. So it cannot be controlled, I would say, because of the needs, like how it's been need. Once we are self-sufficient, then maybe people can try that. So that's what I feel. Maybe there may be a different answer. Uh, so the second one, single cell protein, I'm not quite sure because I didn't work on that. I have no idea on that as well. Sorry.
Online participants, if you have any questions, you can unmute and ask your question.
I was asked by one of the students to know what is the job or study opportunities in abroad. So I may know like those, if you are people interested, I can spend some time on discussing that on Wednesday morning in the biotech department, like as madam has suggested as well. So if that's a thing, maybe if anyone interested, like what are the opportunities, or like study opportunities, what are the fellowship, how you can prepare for that. Um, I was an average student too, so you don't have to worry about like you have to be really, really study hard. So it's not a thing. You just need to be curious enough to grab the opportunities, learn, and then you can do better. So that's the only thing. It doesn't need to be like you need to be very, very smart student. That's fine. So if anyone interested, curious about it, like I can spend some time discussing on this Wednesday morning. Maybe ma'am wanted to contact one in the uh, Plant Biotech CPMB, maybe around like morning time, I guess.
Any other questions from students?
If you want to go abroad, you have to ask question. Not to me, like you had to ask, because definitely I had to open your shell.
Dr. Sambat, this is regarding the population from biparental cross. Because largely scientists do this here. We wanted to emphasize selection. How best this modern technology, otherwise, what kind of selection can be effective to pick a plant of your interest?
Yes, sir. Yeah, that's a good question. A really tough question if you ask a breeder. Like you see a plant, it's everything looks same. Which one I had to select for to advance the generation, right? That's where the genomic selection gonna help. Marker is the one is your handy tool, but how many markers we are surveying? That's also another. Now we have the SSR marker, SNP markers. Yeah, and we don't know, like we are definitely we have that number of markers is quite good enough to survive that trait that we needed. Um, Marker assisted selection is still that's current trend, and genomic selection can help you to select better. It can eliminate the bad ones, so you don't have to use the bad ones to advance it. So you know that, so this is not really performing well. So then you don't want to advance it. So which means that you're reducing that bad ones. You have some good kind of, still you have a good number of ones you wanted to analyze. That's where you can use marker assay selections. For now, marker assay selection is the only one, as I know, has been there, and that markers that's been used, what is been changed. Not only the SNP markers, now we have that structural variant markers, as I told you. It what you can't predict with SNPs, single nucleotide polymorphism markers. You can use structural variant markers to identify the lines. So still marker assay selection, but what markers they are using is, that's the significant, that's the important thing, I would say. I hope that's answered your question. No problem.
I would like to thank the speaker for providing insight on the integration of omic approaches and also the speed reading program. And I request Dr. Kokila Devi, Professor and Head, Department of Biotechnology, to provide vote of thanks.
Thanks.
Good evening to all. So on behalf of CPMB and on my own behalf, we convey our deep regards and hearty thanks to our Vice Chancellor Madam for approval to conduct this endowment lecture. And I'm also very much thankful to our Director CPMB as well as in charge for the Dean School of Postgraduate Studies for motivating us as well as giving the continuous support in conducting this lecture. And our special thanks to Dr. Sambat Parimal. He is all the way from Canada for giving this endowment lecture. And we are thankful for your knowledge sharing as well as for motivating our students for his guidance in career as well as for the higher studies in Canada. And I also thank our senior professor, Dr. John Joel sir, for giving the welcome address in this for this endowment lecture. And I'm also thankful to our professor and head biochemistry, Dr. Uma Madam, as well as other senior faculty members, Dr. Arul sir, Rajagopal, and others like Dr. Saranya. She is the keen to organize this endowment lecture, as well as Dr. Jayakanthan, Bharati, and other bioinformatics staffs, Dr. Krishnan, SRF, as well as Caroline, in organizing this event, a more successful one. And I'm also thankful to our coordinator, Dr. Raghu, in coordinating our students to attend this endowment lecture. I'm also thankful to the coordinators of plant physiology, Dr. Ravichandran, as well as Dr. Rajeshwari Madam of plant breeding and genetics for sending their students to attend this event. I'm also thankful to the online participants as well as and others who are directly or indirectly involved in conducting this lecture more successful. Thank you once again. Thanks for the opportunity given.